Hey y'all, how are y'all doing today? Welcome to your lesson in the chemistry of respiration in the body. First, let's start off with some basic anatomy of the body to understand how the process will work. There are several organs and several organs and muscles within the body, and also bones that work with the respiratory system. Let's start with the organs. First, you also have your oral cavity, which is the mouth, and then you also have your nasal passages. These nasal passages in the oral cavity go down and form the trachea. The trachea passes through the larynx and the pharynx, which both filter the oxygen and the air that comes in, so that you come out with mainly oxygen and not all the other gases in the atmosphere. And then from the trachea flows down to your chest to about this area right here, where it branches into two tubes called bronchi or bronchi. These two tubes go into the left and the right lung. And then from there, these two bronchi split into smaller branches and even smaller branches called bronchioles. These bronchioles then are attached or have sacs attached to them called alveoli. These alveoli is where the main process in all of the chemistry of the respiratory system occurs. Now that we know about the anatomy of the body and the respiratory system, let's start with the basic chemistry and how the airflow works in the respiratory system. So first, the air is taken in through the mouth and the oral, which is the oral cavity, and then also through the nasal passages, which is the nose. The air flows from the nose into the trachea, and the trachea goes through the larynx and the pharynx, which both filter out the air and to get a more pure form of oxygen. Then the oxygen continues to flow down to the trachea into two tubes called the bronchi, which are located about center chest. These two tubes, called the bronchi, branch into smaller branches of themselves called bronchioles within the lungs. And then within the lungs, you have the alveoli. The alveoli take the oxygen into them and also take deoxygenated blood from the pulmonary artery. Earlier, I mentioned a process called diffusion. Now, diffusion is defined as the movement from of a gas or a liquid from an area of high concentration to an area of low concentration. So what this means in terms of respiratory system is that when you breathe in the oxygen and it moves to the alveoli, you are also having deoxygenated blood carried by the pulmonary artery, also brought to the alveoli. Now, since there is a high concentration of oxygen in the alveoli and a high concentration of carbon dioxide, the waste gas produced from the body, and the deoxygenated blood, and a low concentration of carbon dioxide in the alveoli, and a low concentration of oxygen into the pulmonary artery where the deoxygenated blood is, the two gases basically switch places, and the oxygen binds to the blood and is carried back throughout the body to provide the body with the oxygen that is needed, and the CO2 is replaced into the alveoli and then pushed back up through the bronchioles into the bronchi, up the trachea, and then out of the mouth or nose. This is how the basic process of diffusion occurs. The binding of oxygen, or O2, to hemoglobin is a very complex process. First, let's start with the hemoglobin molecule. The hemoglobin molecule has 4 bonding sites on one molecule that allow for 4 molecules of oxygen to attach to that molecule of hemoglobin. And then within one red blood cell, there are approximately 250 million molecules of hemoglobin. However, there are some hemoglobin molecules that do not bond with oxygen. These are ones designated for CO2 or other waste gases that remove the waste and carry the waste from the body to their respective waste disposal systems. And then approximately 1 gram of hemoglobin will be able to bond with approximately 1.5 milliliters of oxygen. And so that means that in 100 milliliters of blood, which has about 15 grams of hemoglobin, 20.1 milliliters of oxygen will be able to bond with that 100 milliliters of blood. The O2 bound to hemoglobin in relation to partial pressure of O2 is shown by something called a dissociation curve. This shows that at 100 torr, all bonding sites on a hemoglobin molecule will be filled. However, for the bonding of oxygen to hemoglobin to occur, there are some standard conditions, temperature, pressure, and pH that need to be present for the process to happen. Temperature needs to be 37 degrees Celsius. The pH of the surrounding area needs to be 7.4, and the partial pressure of carbon dioxide in the body needs to be 40 millimeters mercury. Now we're getting into the real chemistry. I'm about to talk about the bonding of oxygen to the hemoglobin in the red blood cells. So for the experiment I did to represent the process of oxygen bonding to hemoglobin within the red blood cells, it's pretty simple. It's basically I took 50 milliliters of a copper sulfate solution, which represents deoxygenated blood. I poured this into a test tube about almost halfway. From there, I added 15 to 20 drops of household ammonia. This represents the oxygen being placed within being mixed with the blood. From there, a blue clear precipitate will form. This is basically the start of the bonding process, where diffusion occurs and the oxygen is taking place of the carbon dioxide, and carbon dioxide is taking place of the oxygen, and the gases start to change places. And then from there, I went and added 15 to 20 drops of hydrogen peroxide. This is signifying the bonding process. This chemical reaction will produce a large amount of foam, which from there represents the full amount of oxygenated blood that will continue to pass throughout the body. The next part of the experiment you're about to see is what happens when carbon dioxide is taken in with oxygen. Obviously, there's not going to be as much oxygen because there will be carbon monoxide with or in place of the amount of oxygen that should be there. So again, I took the copper sulfate solution that represents the oxygen in blood, poured it into a test tube, filled it approximately halfway. From there, I added less than 10 drops of ammonia, and the ammonia again represents oxygen. Now, since I only added less than 10 drops, this means that carbon monoxide is with the oxygen or ammonia and is going to block the process from oxygen in the blood from forming. Again, a blue clear precipitate formed, however, it was much smaller. And this is the start of the affected bonding process because there is carbon monoxide in there, so that means less oxygen is being pushed into the red blood cells. From there, I added the same amount of hydrogen peroxide, 15 to 20 drops. This is the start, or this is the ending and middle of the affected bonding process, and very little foam was produced. A dark, cloudy precipitate was formed, and basically what this represents is the oxygen and carbon monoxide bonded with the red blood cell, and this let's say the result doesn't look as good as the foam did. It looks bad. It looks like something that you wouldn't want in your body. And this is basically what happens. You get carbon monoxide in your body, and it will start to wither away the muscles and the organs and the tissues that need oxygen to survive, because the oxygen needed, or the full amount of oxygen needed, isn't getting to the body. This whole experiment and all the research is done as a PSA about smoking. Smoking kills. Smoking has consequences, and the biggest consequence of smoking could be your life. Think before you smoke.